know that. But I am an enthusiast, and like so many of you, it's my interest that drives me to learn more about things. So I thought I would step back and share a little bit of what I've learned about the North Texas mushrooms just by being out in the field as a naturalist. So that's where I'm coming from with this presentation. I thought I would start with something that everybody should be aware of, and that is Texas has a state mushroom, and it is called the Texas star. It is, people are reporting it right now. So November seems to be the peak season for these. They're really quite cool. Um, it's special because it's only found in two places in the world. One is Texas, and the other is Japan. Um, its common name is, its other common name is called the devil's cigar. And that's because it comes out of the ground looking like an old stogie, if you know what, if you know what a stogie means, a little cigar. And then it splits open into this star and releases its spores. Like I said, right now is the best time to go find these. You're going to find them underneath of cedar elms. That's their partner. That's their tree partner. So we have a lot of cedar elms, so you could spend some time looking around. They might not all be as beautiful as this one that was photographed for the Fort Worth Botanical Garden. They might look kind of mushy and black, but they are out there. What I think we ought to do is I think we ought to lobby Mary Pearl and Michelle Haggerty to have the Texas Star be one of our, upcoming annual certification pins. I think it'd be really pretty cool. So let's go ahead and get started. So the way I've structured this presentation is kind of into three sections, and I'll pause after each section so we can take a question or two. But basically, I'm going to go ahead and walk through the life cycle of mushrooms just to refresh everybody. I'm sure many of you learned this at one stage or another in your lives, but we'll go through it one more time. And then I want to show you a handful of interesting fungi that you can find right here in North Central Texas. I wish we had 2 hours because I could give you 2 hours of mushroom talk, but we're going to have to keep it to at least one. But anyway, then I'm going to finish up with some tips on how you, as naturalists who might be interested, can get out in the field and look for mushrooms and give identification a try. Okay, so let's start with the life cycle. Setting aside things like lichens and molds, which are related, we're going to focus just on the fungi with the large fruiting bodies that we think of as mushrooms. Some people call them toadstools. Spores are actually the reproductive cells that move away from the mother fungus on air currents. Or by mechanical means like the force of a raindrop, spreading, causing the spores to jump into the air and move away. Or animals, sometimes spores are carried by animals, including us. But here's an amazing fact. A single mushroom is capable of releasing an incredible 1 billion spores per day. That just kind of blows my mind. And they do that over the course of several days. So there's a lot of spores out there, but of course, not all of them are going to grow and inoculate the substrate and then grow. But they're very tiny. You can fit 25,000 mushroom spores on the pin of a head. That's how tiny they are. Okay, so when spores fall onto a favorable substrate, they're said to have inoculated it. So that's a term you're going to see. They can lay there dormant for years, just waiting for the right conditions to grow, to germinate, which of course is going to include suitable moisture, and that is key. So at germination, the spore begins to grow this network of fine little threads. They're microscopic, of course, and they're only one cell wide, but they're called hyphae, or hypha is the singular. And these hyphae are what creep over the food, over and through the food. Now, when a hyphae from two different mating types come into contact with each other, they fuse, and then the resulting hyphae contain the nuclei of both of the mating types. Now, here's a fun fact too. Um, Fungi can have thousands of mating types. Uh, that is something else that blows my mind. It's not just male and female, they can have thousands. Um, so don't let that little plus and minus sign on the slide fool you. So this is the sexual reproduction. By the way, fungi can also produce asexually. Just depends on what the conditions are. Um, so as the hyphae continue to branch, they're going to start branching. Um, they will grow and they'll form what is collectively known as the mycelium. And I know most of you have heard that word, mycelium. The mycelium is essentially the vegetative body of the fungus. It's what's doing the work of finding food and growing. And depending on what kind of fungus it is, they may be reciprocating with another organism. So here's a picture. Of course, this is a microscopic picture of mycelium. Over the course of several weeks, the mycelium grows, and it grows into this tangled mat. The hyphae release chemicals that actually dissolve the food, and the digested nutrients are then absorbed by the growing fungus. So that's kind of the simple way that it works. In this microscopic view, you can see the whole network. But remember, hyphae are just a single cell wide, so you won't see them with the naked eye. The thicker, darker strands that are in this particular photograph are actually ropes that sometimes form when hyphae intertwine. Okay, so as the mycelium expands, eventually it'll have enough mass to create one or more hyphal knots. Now, the knot is sort of dense, if you will, and it will release enzymes into the substrate that optimizes the environment for growing the fruiting bodies that we think of as mushrooms. So this is like the primordial, the primordial fruiting body. The whole process takes place out of sight in the substrate, and it takes a few weeks to complete. But what results is called a pin. And those of you who might have purchased kits where you actually grow mushrooms, you'll be aware that when they do start to grow, you'll start to see pins on the substrate, and those turn into your whatever it is that you're growing, oyster mushrooms or what have you. But really, these tiny little proto mushrooms are just similar to a compressed sponge that can expand very rapidly when it encounters water. So this is interesting too. Most of the cells in the fruiting body are already present in the pin. There's very little cell division that goes on. That's pretty incredible. Okay, so this is just kind of a diagram of the development of a toadstool, if you will, a mushroom that has a cap and a stipe. But it's interesting, not all of the pins are selected for growing into fruiting bodies. Some of them just never progress any further than being a pin. But the mycelium is going to start pumping water into the pins and cause the mushroom cells to actually inflate. And this can happen quickly. I'm sure you've seen mushrooms in your own lawn and garden. They just like sort of pop up overnight. So in figures A and B, they show cross sections of the mushroom. And you can see that, yes, nearly all of the mushroom structures are already in place. But of course, it's going to continue to grow and expand, and spores are going to develop. And guess what? When the spores are fully mature, they're released and the whole cycle begins again. So that's a quick flyby of the process by which mushrooms and fungi grow. Now, it's going to be different for different species and different families and different orders, but for the ones that we usually think of when we go walking in the woods, this is what happens. So, okay. So with that, what I'd like to do is walk through some of my favorites that I've encountered out in the woods, and just right here in Collin County. I think probably some in Denton County as well. And it's now really pretty good time to look for some of these. One in particular is this thing called the split gill mushroom. Um, there are very few mushrooms that have, or fungi that have a worldwide distribution, and this is one of them. It's the most commonly reported mushroom in the Metroplex, and I'm sure you've encountered it on dead limbs or dead trunks of trees. Especially after a rain, they will start to puff up and they'll be a lot more visible. So maybe tomorrow would be a good day for a walk in the woods. Um, if you look closely at the underside of this mushroom, and that's what we're looking at in the large picture in the center, um, it might look like there's a single gill when actually there are a pair of gills. And if you look real closely towards the edge of this one near the bottom of the slide, you'll see that there's actually a pair. The spores are actually produced on the inner surface of each gill slit. So there's like two, the slit is formed by two membranes, and the inside of those surfaces are where the spores grow and mature. Um, when moisture is plentiful, like tomorrow, um, those slits will open and spores will be released. So the reason that scientists think these slits were evolved in this particular mushroom was a way to allow the slits to close up in times when there wasn't sufficient moisture. And you can, in the dead of summer, when it's really dry and droughty, you can find these. They look pretty shriveled up and kind of hard, but they're just waiting for moisture. Now, here's something I just learned today, actually, and I thought this was pretty interesting. It turns out that if you walk in the woods at night and you have a UV light, these things glow blue. And there's a little picture down in the lower left from Alan Rockefeller. Beautiful photograph, but hey, it might be fun to look for these on a night hike if you have a battery powered UV light. Okay, this one is awfully pretty. It's called the flower pot parasol. And some of you may have encountered these. I know that at least one person on this call has encountered it recently. Um, it is something that frequently will pop up in a flower pot. And the reason for that is it's common to the tropics, which makes it pretty well suited to greenhouse environments and indoor planters. So if its spores are around and make it into a communal pot or commercial potting mix, they may end up in your flower pots. Or if you had a flower pot that had some spores and dumped that soil out into the yard, you may find them there. This mushroom has an alkaloid called birmomins um, that are more or less unique to this species, and they're thought to cause that beautiful yellow color that you see. They're really pretty. Now, one thing I want to warn you, you and your kids and your pets don't want to eat these mushrooms because they are toxic and they will cause severe stomach upset. But as long as you don't eat them, there's nothing wrong with them. You can handle them. They're pretty harmless to people and plants. And they're really quite a novelty. Okay, this one is, I just love these. You will see these, depending on the species, you'll see these in the spring through the fall, and always after after a couple of rains and moist conditions. And this is a woodland fungus. It's called a wood ear, and some people call them jelly ears. We have a couple of species here in the Metroplex, but they're called a jelly ear because when they're moist, this one's a little dry, but when they're moist, they do look like jelly, and they do look like ears. Um, they have wonderful wavy lobed margins, like you can see here. Interestingly, unlike a gilled mushroom where the gills are kind of pointing downward and the spores fall out of the mushroom that way, these actually have their what's called the fertile surface, the surface that has the gills, on top. The underside of the ear is actually called the sterile surface. So those are two terms you're going to see when you start reading up on mushrooms. You see them growing on trunks and trees that are still standing but are obviously dying. So that's what you see on the slide. I've given some indication that each of these mushrooms has a preferred medium. This mushroom is saprobic, meaning it feeds on dead wood tissue. Okay, so that's what that means. One thing to note on this and any other mushroom is that they will change their appearance as they lose moisture. So they don't, except for the wood ears, a lot of these soft mushrooms are kind of ephemeral, and they do look very different from one day to the next. So if you're trying to identify mushrooms, that can complicate things. So I just wanted to plug that in there. Now, there is an edible form of auricularia that is cultivated in Asia for food, and some of you may have eaten wood ear in some Chinese dishes. I know I certainly have, and it's delicious. But those are other species, and I'm not going to talk about edibles in this presentation. I absolutely not qualified to talk about edibles, but this is one you will hear of, and it is commercially produced, a different species. I just wanted to note too that there is one species of jelly ear here in north central Texas that if you see it on a eastern red cedar, that will pinpoint the species of that particular wood ear. There are several others species that, you're not going to get that help with. Okay, this one's kind of ugly and shaggy, but it's really super cool too. It's got a lot of texture to the top of the mushroom. It's called hairy hexagonia, and it is a polypore. So if you hear the term polypore, what that means is like a shelf fungus. It grows on wood. When it's dry, it's kind of very hard. People don't eat these things because they're just too hard and too chewy, and who knows what's in them. But they're not one of the choice edibles people go looking for. This poor organism has been reclassified and renamed at least 17 times. Prior to its last reclassification, it was placed in the Hexagonia genus, and hence the common name. They have a pretty rigid structure and shelf-like experience. They are common to the tropics of the New World and the southern part of the US. So if your travels take you to visit relatives in the northern part of the country, you're very unlikely to encounter these. They're kind of special to where we live. They have this dense, kind of hairy stuff on top of on top of the shelf. I call it a shelf. And it's hypothesized that these hairs actually might help retain moisture in a microclimate, which is kind of interesting. You can find these in our woodlands all year long. I've seen them in every preserve and park I've been to in the Metroplex. Now, if you do dislodge one of these, and it is kind of tough to do that, they're attached pretty stoutly to the limb, and turn it over, this is the fertile surface of the hairy hexagonia. And you can see it's made up of pores. Those are actually the openings of long tubes that go into the mushroom. And it's the inner surface of those tubes where the spores are carried and where they mature. And when they're ready, they fall out of those pores that you see there, and then they're spread on the wind. This is a real pretty little one, and it's in the bolete. Well, this is in the Hortipolis species, but it's called a bolete. There's a whole family of mushrooms called boletes, and very often they have the red caps like you see here. They can be very pretty, sometimes not so much. They also, as you can see, have pores on the fertile surface. This is a very fresh mushroom. This is just beautiful mushroom. Um, um, Oh yeah, I'm just looking at my notes. One of the really interesting things about boletes is they are actually mycorrhizal partners with trees. So you will find them growing beneath trees of their preferred partner species. So if you want to go looking for them, it would pay to look up which species are around and what their preferred tree is. Some boletes are very picky about their partners. There's one called the ash bolete that's not quite as pretty as this, but it prefers ashes, and you may be able to find that around, in our woods. These guys make their appearance spring through fall, and you'll notice that if you look in iNaturalist, for instance, you'll see there's a peak right around late September, and they can be quite colorful. Sometimes they're bright red. I just love them. If you go to Leela Nature Preserve and walk the Blackjack Trail after there's been sufficient moisture, these guys pop up all over. Pretty interesting. Sometimes when you see them in the wild, they will have little bits and chunks taken out of them. That's because snails, we were talking about snails earlier, snails and slugs like to eat them. So you'll see little chips, and there's insects that eat them. And for the birders out there, it turns out that the brown thrasher has a preference for eating boletes. So who knew? This is a really cool group of mushrooms. They're called stinkhorns. I just love that. Stinkhorns are amazing mushrooms. They are notorious for just popping up suddenly and unexpectedly in anywhere in suburban settings. They come in an array of shapes and sizes. They all look like horns, and they're all pretty colorful. Um, you got to love their colorful names too. So here's a couple: wrinkly stinkhorn, stinky squid, lizard's claw, devil's dipstick, and flasher. just just to name a few. They're pretty notorious. And yes, they are stinky. They are truly stinky. Some part of the fruiting body at some stage of the development will be covered with the foul smelling slime, like you see on the devil's dipstick on the right. That one's pretty fresh. And that slime has a reason. It puts us off. It smells, sometimes it just smells awful. Sometimes people think it smells like dung. However, it does attract blowflies, beetles, bees, slugs, a lot of different insects. And the idea is that the invertebrates get into that slimy material at the tips of the stinkhorn, and then they carry the spores to other new locations. So that's how these guys get around. They're very short-lived and will naturally disappear quite quickly. I mean, I've had them in my yard and they're literally here today and gone tomorrow. Um, and so I was looking at iNaturalist, and judging from the data there, these generally will peak in the warm months. I've had them come up in mulch in my yard and in leaf debris under, some of my shrubs and stuff like that. But of course, they're dependent on moisture, so the moisture's got to be there. Uh, Now this one is unique as well. It's kind of ugly, and it's so easy to overlook when you're out on a hike in the woods. It's called, the common name is carbon balls, and it's actually a complex of species called Daldinia. Um, the first time I saw them, I thought they were, and they were on an old fallen log that had lost all of its bark. That was at Oak Point Preserve, by the way. It looked like there was black mold growing all over the top of this log, but it actually wasn't black mold at all. It was an accumulation of black spores that had been ejected by the carbon balls at night. Now, this species is very hard and very carbonaceous, and it grows to about 3 inches in diameter, which makes them look very much like a lump of coal. But I have seen them when they were young, and they're sort of a brownish purple color. So you may see a mix of the more mature ones and the younger ones together on a branch. They actually have tiny little bumps all over their surface, and that's where the spores are released over time. You see it in the inset here, you see a cross section of one. Excuse me. If you cross section it, you'll see that there's concentric circles there. And each one of those ridges represents a growing season, right? Now, that's not going to be fall and spring. That's going to be when the mushroom was growing and there was sufficient moisture and all the conditions were right. But the one on the outside is the one that contains the spores that are going to be released. And you can see it's sort of shiny in this picture on the outer edge there. Now, carbon balls do not rot very quickly. In fact, they dry out and they can become much lighter. So I read in one place that Stone Age people would go out and they would gather these and use them as tinder for starting fires, because when you break them open, they take a spark pretty readily, assuming that they're dry. And they apparently burn slowly, so sort of like a charcoal briquette. Apparently they have a particularly pungent smoke. I haven't tried this yet, but I really need to. So I may just give that a try sometime. So don't overlook the ugly ones in the woods. They are equally as interesting as the beautiful ones. And speaking of which, here's one that I think is really kind of cool to look at, very pretty. This is called juniper apple rust. And it's got a very interesting life cycle, and we have all the necessary ingredients in our area for their life cycle. So I don't know if you've ever noticed in the spring, you may see these sort of tentacled jelly-like reddish, orangish, yellowish things growing on eastern red cedars or juniper ashes. It's actually a rust, which is a fungus. Um, Rusts are parasitic fungi, and they consume living rather than dead tissue. So you'll find them on the tips of growing shoots. Let me just get a sip of water here. Um, if you have a lot of eastern red cedars nearby in your yard in the spring, go out and look, and you may see them covered with this stuff. Okay, so um, this needs two species to complete its life cycle. It needs junipers, and it needs something in the prunus family. A pear, an apple. I think those are both in prunus. Anyway, a pear or an apple. And we've got lots of Callery pears in our suburban yards. So they can find their partner. So the idea is that in the fall, the fungus forms this sort of gall-like structure that you see on the inset here at the lower right. And in the spring, those gelatinous horns will begin to form off of that gall. And it takes an incredible 4 to 5 hours after the first rain for these horns to release their spores into the current. It's such a quick process, so you have to be on your toes and you have to be looking for them, or be outside a lot. That helps too. Um, Now, if there's an apple tree or a pear tree close by, say, within a half a mile, um, the spores may land on the young leaves that are starting to grow on those trees and germinate. So the resulting infection, because these are parasitic or pathogenic, the resulting infection looks like yellow spots that turn into blisters later in the summer, um, when it's their turn to produce spores. And when they produce their spores, they get released and off they go to a nearby juniper where they'll germinate and produce another gall. And so the cycle goes. So that is pretty interesting. All right. And this is my second to last one. I don't think I showed this one the last time, but I decided to show it because I just think these things are so gorgeous in a crazy way. So this little guy, he wins top awards for being about the ugliest mushroom around. It has a variety of colorful names like dead man's foot, dead man's fist, and lots of variations on dog turd fungus. So yeah, people notice these things. But for all its ugliness, it actually does perform a pretty valuable service, which it's prized by gardeners and foresters for. And that's because it forms mycorrhizal relationships with plants. So you may be familiar with that term. A mycorrhiza it's really just a symbiotic association between a plant and a fungus. Um, the fungus colonizes the host plant's roots. You may have heard of this. And they exchange what they both need to thrive, basically. The fungus produces, gives the plant phosphorus and nitrogen and water. And what the mushroom gets back in return are carbohydrates and even lipids that help it grow. So foresters will, and others as well, maybe nurserymen, often inoculate the roots of tree seedlings with the dieball spores to give the saplings a boost at growth, right? To help them do better. Okay, here's a fun fact. The dieball, as its name implies, can be used for dyeing wool. It seems counterintuitive, but most of the bright, pretty colored mushrooms are comparatively useless when it comes to acting as a dye for fibers, but not the dye ball. It actually produces a real pretty dark rust color in wool. So I don't know if we have any fiber arts folks, enthusiasts in the audience, but that would be an interesting thing to try, to dye some wool. Now, these will pop up just about anywhere. I've seen them on my walk, like my daily walk in the neighborhood. They look like potatoes. Just look for something that looks like a potato. Okay, here's the last one. These you might be able to see right now. So these are going to be doing their thing in the autumn, and you'll find them on debris that's left on the soil in woodlands, or I find them in my garden, in my front garden, underneath of, leaves that might have blown in and so on. Um, they look like bird's nests, right? Hence the name, bird's nest fungus. Um, you can see in the inset how tiny they are. Those are ones I picked up and held in my hand. They're super small. Um, you'll find them on moist decomposing matter, including dung. So if you have horses or cattle, look on the dung. Um, some of you with livestock might have already encountered them. You stand your best chance of finding these in the fall, like I said, after we get a bit of rain. So get on out there this week. The so-called eggs that actually sit inside this fungus are actually lens-shaped capsules called peridioles that contain the spores. And they rest inside that little cup. And then when it rains, a splash of rain will force that little capsule to pop out and land somewhere. And as the spores mature inside that capsule, the capsule will pop open itself and release the spores into the air. So they are pretty cool. Yeah. So that was a run through of some of the mushrooms that you actually can encounter out there, and it's not that hard to find them. I'm going to, let's see, I'm going to open chat and see if there are any questions. Okay, I'm going to move on to our next section. Now that you know some of the things to look for and you know some fun facts about them, might be a good time to hit the trail. So I think I took this photograph at Leela, if I'm not mistaken, on one of probably the Blackjack Trail near the stream. That's a great place. All right. One thing, as master naturalists, we all need to be aware, and I'm sure we all are already aware, but I'm going to reiterate it anyway. We need to get permission if we're going to be gathering anything off of someone else's land. So there are a lot of foragers out there in the Metroplex who are enthusiasts looking for edibles. And many of you may have heard of Meriwether's Foraging. It's a website and probably a Facebook page, and gosh, I don't know what else, run by Mark Vorderbruggen. He's Mr. Meriwether, and this is what he says. I'm going to read the quote. Foraging for mushrooms in Texas is illegal on private and public land unless you have permission. And while some national parks and preserves in Texas allow for mushroom foraging for personal use, and that's usually defined as less than a gallon, harvesting fungi that will be sold requires a commercial permit. And this practice is strictly prohibited in Texas parks, and the state regularly assigns hefty fines to violators. So these are the same rules we're all familiar with respect to gathering plants, seeds, whatever you find out there in our local preserves. But just wanted to tell you it does apply to mushrooms as well. Okay, with that said, what I want to talk about is, like, as a naturalist out there in the field, what do you do to identify a mushroom that you find? You're walking along, you see something interesting, you bend down and like, holy cow, it really is a fungus. It's not just a rock. So I'm going to walk you through what to do. Here we have encountered a bolete. Remember that slide I had that had the real beautiful orange and yellow bolete? Well, this is a different one. I think this is the ash bolete, actually, if I recall the species. It's a little more mundane looking, but it's very chunky. It's very heavy. It's got a very thick stem, or stipe, as it's called. You're going to want to do, you get out your number one tool, your number one naturalist tool, and that is your camera. Take pictures of it. Take pictures from every angle that you possibly can. And guess what? It is okay to dig up the mushroom, lay it on the ground, take the photographs. You are not going to hurt anything. This is the fruiting body of the organism, which is out of sight. And you know what? Especially if there's more than one, there might be a cluster of them there. It's okay to pick one up and look at its angles. Just leave it there when you're done examining it. Okay. Use tool number one. Now, we all, I'm sure everybody on this call has used iNaturalist. Some of us a lot more than others. Some of us are kind of gaga over it, and I'm in that camp. But it is a great way to register the fungus that you found and perhaps even get an ID on it. Now, iNaturalist, I have to tell you, it is not as good at giving you a firm ID on a fungi compared to, say, like birds or plants or herbs, what have you. And a lot of that, there's several reasons for that. One is that there's just not a lot of robust reference sightings that have been logged to train the computer vision what a fungus might look like. Secondly, there are very few mycologists and mycophiles that are using iNaturalist and going in and really trying to make IDs. And I'm going to give you a statistic here. I went in yesterday and looked at in the Metroplex how many observations there had been made and submitted for fungus, and how many had been actually gotten um, seconded. I forget what the name of the word is. Oh gosh, I'm losing my mind. But anyway, accepted as the species was identified. And only 26% of fungi in the Metroplex have been ID'd. So that tells you something, right? In my own case, I looked at my numbers, my own personal numbers, and I was down to 20%. So just so you know, you may not get a good ID. And this is true for all of the mushroom apps that are out there. And I've got some pictures here of different mushroom apps that are available for your phone. You're not going to get much better. So if you're after an ID, just take that into consideration. But go ahead and log your fungus anyway. You know, you are contributing to science with observation. So be sure to do that. Okay. Now, you can't, with fungi, you can't beat having field observations and taking notes. Try to be methodical. Start by making some notes about the environment and the conditions where you found the mushroom, because if it's a mycorrhizal fungus, you know, it might be good to know what trees were around, right? What substrate the fungus was attached to. Was it attached to a root? Was it attached to a dying limb? Those kinds of things are important to helping with the ID. Some species prefer conifers, some prefer deciduous trees. Note that. If it's on hardwood, can you tell what the species is? And if you can't tell what the species is, take a look around and see what the predominant trees are. Maybe there's only 2 or 3 species close by where you are. You might make note of that. So field notes are very important to identifying fungus. Photographs just don't cut it on their own. All right, so one way you can study the mushroom and try to take some notes is to detach it. Go ahead, detach it. You can use a knife. You're not, as I said before, you're not killing the fungus. Also, don't worry about poisonous species, because these poisonous species are not poisonous to the touch. They're poisonous if ingested. Okay, so don't worry about handling it. And if you're concerned, make sure you wash your hands when you get home, or carry some hand sanitizer or what have you. Try to get as much of the base as you possibly can, and that you might have to dig a little bit deep to get that, and it might take a little bit of effort to get it detached from the substrate, but try to do that. You can use a pocket knife, or you can use one of these handy dandy mushroom knives that you see here. I like them because they have a curved blade, and they also have this nice brush on the end so that you can brush away debris that might be on the mushroom before you take your photographs. And it also has a little scale on it. You know, if you want to take some measurements, that's always helpful too. Once you've done that, once you've got it up, get your tool number 1 out again, that's your camera, and start taking pictures from every single angle that you can. You want to make sure you photograph everything you can about that mushroom. Then, and I'm going to talk about this in a second. Well, I'll just hold that thought. You can also, if you're going to make a spore print, let's say, like when I'm on a camping trip, I will make spore prints right there at my campsite. I can take the whole cap, or I can take a piece of the cap and make the spore print. So that's always possible too. we'll talk a little more about that. Here's another tool, great to have, and many of you probably already have an array of um, uh, not microscopes, but lenses, handheld lenses for looking more closely at structures of plants and so on. I like the one on the right. I have one of these because it has a light built in, and all I have to do is press the button. And if you're out in the woods on a murky day, this can help, help you get closer looks and actually be able to see the features with a little light put on them. So get yourself one of those, carry it around if you're not already doing that. And then one thing I'd recommend is that you need to know what the structures are that you should be looking at on your mushroom. So take some time before you go out in the field and learn what the morphology of mushrooms is. There's really only a handful compared to plants. This is way easier. Take a gilled mushroom. That's what we're looking at right here, a gilled mushroom. And then starting at the top, you have the pileus or the cap. We often call it the cap. Which on gilled mushrooms is also the sterile surface of the mushroom. So the spores are going to be on the fertile surface. The cap is the sterile surface. Underneath the cap is what's called the hymenium, or what I've been calling the fertile surface. Does have a scientific name, the hymenium. So if you want to be in the know, use proper terminology. This is where the gills are going to be attached. Now on boletes, remember the ones that have the and polypores, they have the pores and the tubes. They actually have tubes. So if you cross section the cap, you will actually see some of the tubes. So that's always a fun thing to do and to take photographs of. Now coming down, we're going to talk about the annulus in a moment. Let's just talk about the stipe. So the stipe is the proper name for a mushroom stalk. But you can call it stalk, you can call it stem, that's perfectly fine. And the vulva is like the little cap that the mushroom sprouts out of. And I've got some pictures that we can look at in a moment. So you're going to want to note, what kind of fertile surface are we talking about? So look underneath the mushroom, try to find the sterile surface. And it's not always easy, believe it or not. There's so many species out there. Here's some pictures. The top one with the yellow, those are called teeth. And the spores are actually released from the outer surface of each of those teeth. We've talked about gilled mushrooms, so you've got that in the lower right. And then polypores and boletes have the pores, which are the openings to the tubes. So just so you know what that looks like, I just wanted to make sure it was pretty clear. Okay, now veils. Veils are a really important part of the mushroom too, and can really help in identification. So you want to make notes about these and you want to take pictures. So what the heck is a veil anyway? Well, let's start with the universal veil. There's two veils. The first is the universal veil. And if you look at diagram A on the left there, you'll see it's actually the protective cover, that layer of tissue that completely encloses the developing mushroom. And at this stage, you might mistake it in the field for a small puffball, right? If you move to diagram B, this is where the universal veil is rupturing and the mushroom is starting to grow. Remember, we talked about the dry sponge, and the mushroom is going to start to just suck in moisture and it's just going to expand. That universal veil is going to break and fracture. And that's what those little white squiggles in diagram B represent. They may disappear. The veil, in some mushrooms, the universal veil completely disappears. In other mushrooms, pieces and portions of it stay attached to the cap, and we call those patches. So, you can think of some of the, well, some mushrooms actually do have those, and that's something you would want to note in your field notes. Does this cap have patches and pieces of the veil still attached to it? There is another veil called the partial veil, and that's the one that covers the gills of the mushroom. So in diagram D, that partial veil is still intact. In diagram E, it is tearing away as the mushroom continues to expand. And it can form this cute little skirt here, or it might just completely disappear and you would never know it was there. So that's what, when they say to look for a partial veil, that's what you're looking for. Here's some pictures of some partial veils. The one on the left, you can see the annulus or the little skirt. That was pretty fascinating. I'm not sure what species that is. Here in the center, you can see there is what's left of the partial veil. There's a little bit of an annulus there. Depending on your luck, you may see one and you may not see one. In the right-hand side, the partial veil actually is still, in this particular mushroom, still attached to the outer edge of the gill cover, the outer edge of the cap. And you can also see there's patches left over from the universal veil on the cap. But check it out. When the partial veil pulled away from the stipe, it left a smooth area. And sometimes this will have a color shading to it, sometimes it won't. And when it does have a color shading, that often is because spores have started to fall out of the gills in the mushroom, and sometimes they get attached to where that veil was and cause that discoloration. So it depends on the species. This may be an important thing to note in your field notes or in your observation in iNaturalist. This is one of my favorites. Pinch it, bruise it, slice it, rough it up. And here's why. So, did you know that some mushrooms actually produce latex? Like this little indigo milk cap on the left-hand side. There are others. So one way to see if a mushroom is going to produce latex is to get your handy dandy knife out and just cut right across those gills and wait for a minute or two, and you may see latex come out of it. I've never seen one of these blue ones before, but I have done this with, what's the family? It's on the tip of my tongue. Anyway, it'll produce a white latex, and it only takes a minute or two. You don't have to wait around for it like you would a spore print. Now, there are other mushrooms like this puffball in the center that if you bruise it, like pinch it, you'll see it change to this brilliant, beautiful, orangish, yellowish color. That's another clue to the species, to what species it is. Now, in this case, it takes maybe 2 minutes for that to happen, and then that will start to fade. So you don't want to just bruise it and walk away and have lunch. You want to kind of stick around for a few minutes and see if it doesn't produce a color. And then on the right-hand side, this is a bolete in cross-section. Boletes are kind of interesting because when you bruise them, they turn indigo, and then that'll turn to black, and it happens pretty quickly, again, within a few minutes. In the cross-section of this bolete on the right, you can actually see, like I was saying before, that the pores are the pores that release the spores are actually at the end of tubes that are the place where the spores mature. So you can get a little view of that there. I'll bet all of you have made a spore print at one time or another, whether that was in grade school or maybe with your grandkids or what have you. But spore prints are so, so useful to the distinguishing between species. You can make prints yourself at home or in the field. Like I said, I do this a lot when I'm on camping trips. And there are approaches and discussions of this on the internet. I'm not going to go through them here. It's really quite simple. You don't need a whole mushroom or a whole mushroom cap. You just need a piece long enough that, or big enough that has some of the pores or the gills. So you could take just a tiny little bit. And what you want to do is, and what I've decided to do is I generally will take a little section. I'll get a piece of aluminum foil, and on the non-shiny side of the foil, that's where I'll place the mushroom. Now, you don't need to put a glass over top of it. Well, I guess if you were going to had a fan in your kitchen and you've got the fan directly on this thing, that you probably would want to put a glass over it to keep the spores from dispersing, but generally just stick it in a place that doesn't have a lot of airflow. And within a couple of hours, you'll have a spore print. This one is kind of interesting, and the reason it's recommended to use foil is because if I had done this, say, on a piece of white paper, I might not have been able to get the color right, determine the color, but you can see it's sort of a light tan color, and that was conclusive for identifying this one. Sawgill mushroom. Okay. Now, there's a lot of these morphological keys that are out there for mushrooms on the internet. Um, I'm just showing you a picture of one here. They can get very, very elaborate. However, find one that you can learn from. And basically, you're going to be looking for features of the cap, features of the gills, features of the stipe, and features of the veils. And I've kind of covered that a fair bit in the last few slides, so I won't go into great detail there. But these morphological keys are kind of nice because they tell you the terms that are used when describing, say, the gill attachment for a gilled mushroom. And you can get kind of good with that. And it's also helpful when you go to use a dichotomous key, for instance, you'll have some sense of what the terms they're using mean. Speaking of which, my favorite website that I go to for trying to figure out what a mushroom is themushroomexpert.com. It's my absolute favorite website. One of the things that the guy who runs this site has done is he has created a dichotomous key that's really pretty useful and pretty simple to use. Now, I happen to know that everybody sitting in this Zoom meeting tonight has experience with dichotomous keys, some more than others. So everybody should know how to use one. And guess what? It works for mushrooms as well. But this is a super good choice as well. Now, I'm going to send out a PDF here once I finish the last couple slides, which we're on, that has some resources. And in that resource will be a couple of field guides for Texas. They have dichotomous keys as well. So check out which ones you like to use. You have multiple resources. Okay. About 3 years ago, about the time I first did this presentation, the North Texas Mycological Association had just formed. They are under the auspices of the Texas Mycological Association. They've been in operation about 3 years. They have a Facebook page that is fun to participate in, so I encourage you to do that. They have a website, and guess what? They have an iNaturalist project. So when you submit a fungus from North Texas, it will automatically be put in. No, actually, I'm sorry. I think you have to join this particular project. So go into iNaturalist, look up the North Texas Mycological Association project, and then in the upper right-hand corner, instead of it saying leave, it'll say join. Go ahead and join it, and then your observations will land there. Now, the reason I suggest you doing this is because they do have some mycophiles that frequent that site, and you might stand a better chance of getting an ID if you're in that project. So, and if anybody needs help with iNaturalist, because I know some of you may be new to it, you can reach out to me personally, or I'm sure if you've been through the training class, I'm sure you know a few people who can help with that as well. So it's a fun thing to do. Okay, last slide. So the Mushroom Expert, I can't give this website enough applause. It's run by a man named Michael Cowell, and he is from Illinois or Indiana. I can't quite remember. He's been working on this. He's a microphile. I mean, he is so into it. And he gives back by maintaining this site. Lots of information here, lots of deep, there's a glossary if you want to know what terms mean. One thing people do with mushrooms is they will try to detect an odor and a taste, and sometimes that will help ID a mushroom. He talks about all that kind of stuff. Also, just like with lichens, for instance, if any of you attended the lichens presentation at the annual conference, just like with lichens, chemical reactions are one way to get positive IDs on a lot of fungus. So he talks a little bit about that as well. Great site, wonderful resource. And with that, I'm done. So close to an hour. And I don't know, here's one thing I want to do, and I don't know if there's any questions in the chat. I really can't see.
There were a couple questions in the chat. Would you like me to read them to you, or?
Yes, please, Kelly,
Yeah, and
thank
thank
you.
you so much, Jean. Okay, so we have a couple questions. And you guys, if you have any questions, like, let us know and stuff. But the couple that came up were if the carbon ball fungus grows for several years.
Yes, it will.
Okay, cool, cool. And oh my gosh, I can't talk, sorry. Is the wood ear fungus usually reddish?
Yes, it usually is. It can be uh, like the color that I showed you there. A lot of times it's
Mhm.
more rusty looking. It's got, a little more orange to the red, but it's along those lines. Yeah.
Wonderful, wonderful.
And I
Hey,
was gonna see. Oh, I'm gonna put a file in the chat. I'm doing that right now. That's a PDF that has resources. Let me just go to my computer. Here it is, the PDF. Getting sent your way now. Um, it's just it's printed double-sided. It's just one sheet of paper, or you don't even have to print it if you don't want to, but lots of resources there if you want to drill into the science behind how these things work or the biology, or if you just want to learn a little bit more about morphology, what have you. There's lots of, I tried to put together a wide variety of resources, so have fun with that. Hopefully that's coming through.
thank you.
Yeah.
Yeah, I saw it, was able to download it. Hopefully some other people have as well. Do we have any more questions? You're getting a lot of thanks and praise in the chat.
Well, thank you guys for joining. Hope it wasn't too long-winded.
No, it was great. It was great. And awesome pictures too.
Yeah, yeah, it's fun. I think they're fascinating. I just like looking at them. Okay. All right. Well, hopefully that was helpful. Reach out anytime. I mean, I'm just one of you guys. I'm learning and, just kind of finding my way through this. So I'm here. You know how to reach me. And if you want to take a walk
Oh,
in the woods and
sure.
look for mushrooms, I'm game.
That sounds fun. Like, should we do like an official thing or, unofficial even?
No, I mean,
Either way.
either way, I'm game.
That sounds great. I don't see any other questions coming through, but you were saying it sounds like people can contact you if they have anything or
Yep.
ooh, we have an idea for a mushroom bio blitz. That could be fun.
Ooh, that would be fun.
Yeah.
Fun. And it might just be the right time of year.
Yeah, after some rain. Oh, yeah.
Oh,
Getting
so
some
we got
people
some
for
people
it.
who would be willing to jump in on that. That's awesome.
Like, well, now, definitely now we gotta.
Oh yeah, one thing I know a lot of people are probably leaving the call, but one thing I didn't mention is if you join the mycological North Texas Mycological Association, they schedule what they call forays. Now they're more into edibles. However, it's a group of people who know mushrooms and want to get out there and look at mushrooms. You could always join one of the forays and maybe you'll have some fun with that. My favorite place to look for mushrooms is just about anywhere. Um, I find them all over the place. So if you're looking for the ones that are attached to wood, then, woodlands are great. Oak Point, the Caddo Trail is great. Blackjack Trail at Leela is great. Any bottomlands would be good for the saprobic mushrooms, the ones that attach to wood. And then the others is, just keep your eyes open. That's the important thing. Keep your eyes open because a lot of them do grow in grass too.
Mm,
Okay.
we had another question. Do you happen to know if wood ears are the same as pig ears?
I'm not familiar with the pig ears being a common name. I'd have to research that. It's not one I've heard. But I think from a Chinese cuisine perspective, I mean, I've been in the Chinese grocery store up here and they do call them, I think they call them wood ears. I've seen them on menus as well, but I haven't seen pig ears. Maybe that's but you know what? Common names, people make common names up all the time. So that could be one.
Yeah, and some like regionally might be different,
Yeah,
something
yeah,
different.
sure, sure. Okay. Well, it looks like things have quieted down there. So
Yeah.
thanks for putting this on, Callie. I appreciate it.
Oh, thank you so much. And thank you like for sharing your wisdom. And it was fantastic. We all
Good,
learned a
good.
lot.
All right.
Okay,
All right.
that was great. Oh, let me
Looking forward to the next one.